The vision of the Wits Research Institute for Malaria is to establish first world researchers with the capacity to reduce malaria transmission and make an impact not only in South Africa but also on the African continent and also at an international plane level. Other researchers, of course, in South Africa in the past that made global impact on the research we do. We think of Professor Hugh Patterson, and he was the first person that sort of expanded our understanding of mosquitoes, the species complex, how they evolved. And he developed a species concept called mate recognition species complex, and that is that if two mosquitoes or two species, whether it's insects, cannot recognize each other as potential mates, then you will have an event called speciation.
We always pupae from the balls to separate the larvae. It's important to get the synchronization correct so that they can all come out at the same time. It is done to save time and so that we also don't have to clear adults every single day. It is done by that process by pouring through the water onto the sieve to separate them, put them into a clean bowl of water, and then basically twirling the mosquitoes in there. And then the synchronization is correct and they will develop very quickly.
We have very specialized facilities here. We call them insectary, and where we rear mosquitoes. It's highly labor intensive and very, very specialized. We try to generate an environment that is suitable for mosquitoes from African regions. So we generate an artificial tropical environment that the mosquitoes will be comfortable in. It's unfortunately very expensive to manage and maintain. You need a staff complement that are extremely committed, because this is 7 days a week commitment from the staff and from the students. Apart from that, it allows us to do research that you can't necessarily do in nature or in the field.
These mosquitoes, we collected them from different houses in different 14 villages. And in each of the mosquitoes that we have collected that were possible to dissect, we dissected the mosquitoes and we assessed the different parts before it came to the laboratory for sample genotyping. So the initial assessment was to assess the mating status of the mosquito through dissections to check the spermathekas.
Over the years, insecticide resistance have expanded tremendously on the African continent, and it's actually now quite difficult finding susceptible mosquito species. So those ones in the insectary are susceptible as well as resistant to insecticides, and they are a very valuable resource, not only for us but also for South Africa and the collaborators we work with as well. And for us to keep on top of the game, we know that we need to have more tools available in our toolbox to actually do vector control. And with insecticide resistance, of course, we try to aim to incorporate other techniques in that's not chemical, so non-insecticidal, that can supplement the existing tools that we have. And for those, we are looking at genetic approaches. And worldwide, there's a huge drive to identify novel targets that we can use to kill or suppress populations, and there's also a big drive towards using genetics so that if a mosquito feeds on somebody with a malaria parasite, that same mosquito are unable to transmit it to another person.
The samples received from other African countries have a combination of secondary and primary factors, and for us to determine the species, it first undergoes morphology, and from morphology, if it's a primary factor, we'll then do PCR. And the PCR process starts from when we dissect the leg or wing of a mosquito. We'll then do DNA extraction, let that undergo a PCR reaction in the thermocycler, and then we'll load through samples in the gel. And that will tell us what type of species it is.
Understanding how the environment impacts on mosquitoes is very vital for us in the research that we do. We need to understand how will these mosquitoes adapt, how will it translate back to disease transmission. So a lot of the work we do is to try to understand and predict what these mosquitoes will do as vectors of diseases.